in all sorts of shapes and forms. And here, for example, we have a sea star larva in the upper left. And right below it is a chitin larva. And over here is a villager larva. That's the larval name given to those larvae that belong to marine gastropods. Right below that is a bryozoan larva. Up here is a tunicate larva. This is a tadpole larva. That's what they're called. And right here, down below, lower right, is a sea urchin larva. And in between those two is a coral larva, also known as a planula. And the coral larva is something that I'll be focusing on, as that has been the focus of my dissertation research. Now, the larval form is a mode of dispersal in the open ocean. So upon encountering a suitable substratum that indicates that this is a perfect benthic habitat for me to plop down and attach myself and settle and metamorphose, I'll grow back into an adult. And as far as terminology goes, a lot of people use these two terms interchangeably, settlement and metamorphosis, but they're actually two discrete processes. And settlement refers to a change in habitat, that is leaving the larval or the planktonic environment and entering a juvenile or the benthic habitat. And this can be a reversible process. However, it is usually followed by metamorphosis, which is a significant change in form from one stage to the next in the life cycle. And this is often associated with a change in function as well. So this involves a loss of larval structures and a gain in juvenile structures. And this is usually an irreversible process. Some major points I'd like to address first. Larvae don't just settle randomly, only where cues indicate that this is an appropriate site for them to grow and reproduce. And larvae of many modal invertebrate species settle in response to dissolved cues from their prey. So for example here in this picture, this is a nudibranch. The species name is called Fistella sabogi, and the adult feeds on Parietes compressa coral specifically. It doesn't feed on any other type of coral, just that one. And in turn, their larvae rely on a soluble cue that the coral produces for them to settle and metamorphose. And it makes sense, right? You settle on the surface where the food source is when you grow up to be an adult. And biofilms, which are defined as matrix-enclosed populations of bacteria that adhere to each other and to surfaces, also provide cues for many other types of larvae, especially those that are benthic species and sessile species. And however, not all biofilm bacterial species are inductive, so not all bacteria are the same. And here's a picture, for example, of some biofilm. So these are some glass slides that have been placed out in the field for 1 to 2 months to develop this nice coat of biofilm. So the bacteria are the first ones to recruit onto these surfaces, followed by other types of microorganisms, such as diatoms, for example. And over time, this biofilm builds up, and those provide the cues for larval settlement. As an overview, the big picture that I'd like to focus on is how do you go from the bacterial cues that induce settlement and metamorphosis to the larva that uses sensory systems to detect it. And I am interested in both sides of that story, and I'll go into both sides of that story, beginning with bacterial induction of settlement and metamorphosis. So what types of bacteria are available on a coral reef, for example, and which ones induce. And this will be followed by sensory systems for detecting settlement cues. So how does the larvae respond to that? And towards the end, this is where I'll tie into some of my postdoc work here, settlement and symbiosis. And in the lab that I'm currently in, we look at another cnidarian model and look at its symbiosis with a group of dinoflagellates. And that's something that I'll tie into towards the end. But first, I just want to get everyone familiar with the structure of a larva. And so structurally going across, it's not very different. But I just want to point out that on this end here, the right side is the oral end because it opens up to a mouth. And on the opposite side, the left end is called the aboral end because it doesn't have a mouth. And the brown pigmentation that you see here are those symbiotic algae, the dinoflagellates, that are living within this host, within this cnidarian host. And the other one I might as well introduce to you now is the other species that I look at in my postdoc. And this is a larva of a sea anemone. So on the left-hand side, it's a coral species. On the right-hand side, it's a sea anemone. And these do not have the symbionts to begin with. So they sequester the symbionts at the larval stage, at a later larval stage. So first, I'll begin with the bacterial introduction of settlement and metamorphosis. The specific questions that I'm interested in are, is there specificity in the inductive capacities of different bacterial species? And does biofilm density play a role? Does it differentially affect larval settlement? And this is a general life cycle of a coral. And so corals reproduce in two different ways. They either do broadcast spawning, which produces gametes, separate sperm and eggs, that then develop into a zygote that further develops into a planula larva, or they can brood their larvae directly. And this requires internal fertilization, and when they release the larvae, they are fully developed larvae at this point. And so the larva then further develops into a polyp through settlement and metamorphosis. So this is the process that I'm interested in here. And then finally, the polyp grows back into an adult over time. So over the past 2 decades, many research studies have focused on settlement cues. And these settlement cues are quite variable and often species-specific for different species of coral. And the two common ones, the most common ones, are crustoscorallin algae and the bacteria associated with crustoscorallin algae. And I'd just like to add, though, that even though there's a whole growth in this field here, looking at settlement cues, what's really necessary is a better understanding of the underlying cellular and molecular mechanisms, physiological mechanisms that are utilized by the larvae to detect the cues. And that's what I would like to constantly try to tie together in this talk here. And for this first part of my study, I looked at this particular coral species known as Postdoctoral Researcher. It's a common Indo-Pacific coral in Hawaii, and they brood their larvae year-round according to phases of the lunar cycle. So unlike a lot of broadcast spawners that only reproduce during the summer months, this one does it year-round. And a typical colony like this, that's 10 centimeters across in length, can release anywhere from 10 to hundreds of larvae per day during that lunar phase, which usually lasts a week each month. And this is what their larvae look like. I've already shown you a picture of one before, but it measures about 1.4 millimeters in length. I said millimeters, so that's actually pretty large for larvae, for a lot of invertebrate larvae that are on the scale of microns. So this is pretty large in comparison to a lot of other species. And when they settle and metamorphose, they change their form essentially by flattening in the oral-aboral direction. And this is what they look like after they've settled. And this is a group of newly settled juveniles. So the aboral end comes down onto the surface, attaches itself, and it flattens. And then you have the oral end, the mouth end, sticking up. And that's how it feeds. And over time, the calcareous base here, the white, has become fully formed. And you can see the tentacles of the polyp peeking out from the center here, and some other smaller polyps that have asexually budded off on that calcareous base. So how do I collect the larvae? Well, first of all, I'd like to introduce to you the larva collector. And these are simple pancake batter bowls purchased from Kmart that have been placed in a sea table like this with running seawater. And the colonies get placed in the pancake batter bowls. And when the larvae are released, they're positively buoyant. And so they'll come up to the surface of the water, flow over the spout there, and get collected in this sieved container, which has a mesh at the bottom. And essentially, when I come in the next morning, I have larvae to look at. So it's a pretty efficient system. As far as how I collected the biofilms, I put out some artificial surfaces, such as glass slides, in the reef to develop biofilm over time. I also collected some natural surfaces from the reef as well, such as pieces of this coral here, Parietes compressa, which is a common reef-building coral, as they're pretty massive. The adult itself, Possilophora damicornis, as well as another reef-building coral, Montipora capitata, and crustose coralline algae and a red alga that's really common and invasive, actually, in Hawaii, known as Gracilaria salicornia. And the biofilms were either scraped off of these surfaces or vortexed off of these surfaces and into 1 ml of sterile filter seawater. And then I would make monospecific biofilms from those. And by monospecific, I mean that they get poured into a dish, and the dish is left to stand for an hour for the bacterial cells to attach to the bottom of that dish. And that creates a single layer of marine bacteria consisting of just that one species. And with that, I was also able to identify these different bacterial isolates by sequencing their 16S rRNA gene as well. So here are the results for those settlement experiments.